A single individual chemical reaction is a multi-step process in which the energy that's required to break or weaken the bonds in a substrate molecule and reach its transition state is referred to as the activation energy. And this is the energy that's released after new bonds or the product is formed. Enzymes help to really drastically lower this activation energy, and they typically do so by a factor of 1,000,000 or more. And so, as I mentioned at the beginning of the video, these reactions could take place. Enzymes just help to essentially speed up the reaction considerably by lowering the amount of activation energy that's necessary. And in this process, if energy is released, it's referred to as an exothermic reaction. If it's absorbed during the reaction, it's referred to as endothermic. As enzymes are proteins, they are synthesized by ribosomes. And we know from past units that ribosomes can be found in two different locations primarily. The enzymes that are used for extracellular work, exoenzymes, they're released from the cell for external work, and they are generally synthesized by ribosomes that are attached to the endoplasmic reticulum, rough endoplasmic reticulum. Oftentimes they're used to break down larger molecules, such as allowing molecules that have been broken down during the process of digestion to pass across the plasma membrane. Ribosomes that are synthesizing enzymes that are floating around in the cytoplasm, so the free-floating ribosomes, they are generally catalyzing reactions that take place inside of the cell, such as glycolysis. That's part of the aerobic respiration cycle that we'll look at in our next unit. In a metabolic reaction, the conversion of energy is never 100% efficient. The products always contain less energy than the reactants, and that energy is converted to heat. That excess energy that's lost is converted to heat, and this is actually advantageous for some organisms, including ourselves. Birds and mammals use this heat to help maintain body temperature, and excess heat in humans, particularly like during exercise, causes the production of sweat, in which we use evaporative cooling to help dissipate and release that heat. But this is helpful because it allows us to be able to maintain our own body temperature. We can see in the thermal images here the birds that contain that have heat, their heat production, and then in the other image, difficult to see, but the glowing oval is a mouse that's being consumed by a snake, and the snake is a little bit more difficult to see because it does not regulate its body temperature. You've probably seen reptiles before sunbathing, and that is them warming up their bodies because the environment dictates their temperature regulation. And so this requires the use of energy within birds and mammals to be able to maintain our body temperature, requires use of energy as opposed to the snake that uses the environment to be able to maintain temperature. When the basal metabolism, which is the energy that's used while the organism is at rest, just in order to maintain itself, when it doesn't release enough heat, then the metabolic rate has to be increased, and this would be the energy used for cellular processes. And so different organisms have adaptations to help them to be able to increase the metabolic rates, to be able to warm themselves up. Birds, a good example would be penguins huddling together in groups to share the heat that's released by their neighbors. So we see them in groups huddled together. Humans, if we get too cold, shivering produces heat by the muscle contractions and helps to raise the core temperatures. And then also mammals have brown fat tissue. And these are cells that contain mitochondria that carry out uncoupled respiration, meaning it allows the production of heat without the production of ATP. These brown fat tissue cells do. Large chemical transformations don't usually occur in a single step, but usually requires multiple steps or multiple small reactions in order to carry out an overall process or metabolic pathway. And that's what makes up a metabolic pathway, is multi-step small reactions put together. And this can be in either a chain or a cycle, a linear chain or a cycle. Glycolysis would be a good example of a multi-step reaction that takes place in the cytoplasm and results in the splitting of glucose, and we'll take a look at in our next unit. And overall metabolism is a network of interlinked and interdependent pathways, these multi-step pathways put together. Branching of pathways is also common.
Again, going back to glycolysis, the molecule, the 6-carbon glucose molecule that it starts with, actually gets split into 2 3-carbon molecules, and then those two each undergo the same steps. So that would be a branching. And so a linear example would be glycolysis. A cycle reaction example would be Calvin cycle, which is used in photosynthesis, and Krebs cycle, which is used in aerobic cell respiration, in order to produce ATP.
As mentioned previously, each enzyme is a little bit different, and many enzymes have a second binding site besides the active site. That's called the allosteric site. And this is where a different substance can bind and unbind. It's not a permanent binding. And if the substance binding blocks the ability of an enzyme to be able to conduct the reaction, it's called an inhibitor. And in this case, the binding, if it occurs at the allosteric site, then it's called a non-competitive inhibitor because it's not competing for the active site. The binding of this inhibitor at the allosteric site usually causes a change to the enzyme, specifically at the active site. And so this then would turn the enzyme on or off, kind of like a light switch, on or off, in that the binding deactivates the enzyme because the active site doesn't fit with its substrate anymore. And so this, again, is known as a non-competitive inhibitor because it's binding at the allosteric site. It's not competing for the active site.
Competitive inhibitors are similar to allosteric or non-competitive inhibitors, yet the competitive inhibitors are actively competing for the binding site, the active site of the enzyme, and when it does bind, it blocks the substrate from being able to bind, thereby turning off or shutting down the enzyme. The competitive inhibitor is structurally similar to the substrate, but it doesn't allow the product to be able to be formed. And a good example of this is found in statins. It's a medicine that works by competitive enzyme inhibition, and it's used to treat high blood cholesterol. A little bit of background information. Low-density lipoproteins, or LDL cholesterol, is considered the bad cholesterol, and it collects in the blood vessels and it causes narrowing, which then prohibits and limits the ability of blood to flow through the vessels. High-density, or HDL, lipoproteins are considered the good cholesterol in that they help to move the LDL out of the bloodstream, so those are beneficial. These statins, the medication, they bind to the active site of an enzyme called HMG-CoA reductase, and this enzyme catalyzes a reaction in metabolic pathway that's used to synthesize cholesterol in the liver cells. So if the pathway is turned off by statins, then there's less cholesterol that's produced. As the collision between an enzyme and a substrate or inhibitor is random, whenever the active site is empty, either the substrate or the inhibitor can bind to it. It's totally random because the enzyme generally and the substrates or competitive inhibitors are moving around randomly as molecules do. The rate of inhibition becomes greater if the concentration of inhibitor increases. So as there's more and more inhibitor, then it's going to increase the rate of inhibition overall. So if there's a low inhibitor concentration and the substrate concentration is increased, then inhibition is going to be reduced significantly because there's more substrate than inhibitor. This is not true with non-competitive inhibitors because the inhibitor is not competing for the active site. So in non-competitive inhibition, the inhibitor is binding to the allosteric site, and so it's not competing with the active site, and so it's not reduced. That inhibition rate is not reduced with more substrate added. Metabolic pathways must be regulated to help ensure that the correct amounts of products are produced, but there's not an excess of those products. If there's an excess, that's essentially wasting energy for the cell. And a way to control or to ensure that excess products aren't produced is a process called feedback inhibition. And this is when a situation in which the product of the final step of a metabolic pathway acts as an inhibitor to the first reaction at an allosteric site. So this is also an example of a non-competitive inhibition process because the product is binding to that allosteric site. If too much of the end product is produced, it increases the chance that the competitor, which is the final product of this pathway, it will end up binding to the enzyme, probably the first enzyme or one of the enzymes in the chain of the reactions, and thereby stopping the synthesis of more of that product. If there's too little end product, then it's going to continue, that pathway is going to continue to producing products because there's a low chance that the product will actually bump into the enzyme and bind to it. It's an efficient method to be able to regulate an overall pathway, and a great example of this is the process of threonine deaminase, in which the enzyme threonine deaminase is used to convert threonine through 5 overall enzymes into the product isoleucine. And when there's a lot of isoleucine, more of it's not needed to be produced, and so that isoleucine acts as a non-competitive inhibitor to threonine deaminase by binding to the allosteric site. And when it does that, it turns off the pathway. It blocks it from continuing to produce more isoleucine, so it's kind of like a regulated off switch. And it's really an efficient way to be able to ensure that excess products are not being produced.
Sometimes inhibition can also have permanent effects, and they can't be undone. Toxic substances such as mercury and lead are examples of non-specific inhibitors to a variety of different enzymes as they bind permanently to sulfohydryl groups in the amino acid cysteine. And this causes the metals to be really toxic and dangerous pollutants because they block those enzymes from being able to use. And so mechanism-based inhibition occurs when the inhibitor is structurally similar to the substrate and it binds to the active site, but it permanently binds to the active site via covalent bonds. And this produces a stable bond that results in the enzyme not being able to work as the catalyst. It's essentially non-functional, causes harm to the organism as the change is permanent, and it can also even result in the death of the organism if the enzyme function is vital and there's lethal levels of the inhibitor concentrations. An example of this can be found with the use of penicillin, which is actually beneficial to us. Penicillin synthesizes the enzyme inhibitor to kill the organism, gram-positive bacteria. Penicillin enters the active site and then permanently blocks that site, and that becomes lethal for the gram-positive bacteria. And so this concludes our examination of looking at metabolic pathways and enzymes and how they function and how they can be influenced or impacted. Over the next couple of units, then we'll begin to look at actual specific metabolic pathways and the process of photosynthesis and cellular aerobic respiration.